Hello, good evening. Welcome to My Abbey Fences, the European Fertility Society. We are back once again with a fascinating topic today for sure. So, thank you so much, everyone, for joining. There are lots of you that joined us today. I'm sure you will have plenty of questions for Dr. Uliana Dorofeyeva. You can see her right here. Hello, Dr. Uliana. Welcome back. We already discussed this before, that it's been a while since you've been here, but I'm really happy and excited to have you back. This is not an easy topic, but I'm sure you are going to explain everything as always. And how are you? It's great to have you.
Thank you so much, Caroline. It's a great pleasure for me to be here. It's been quite a while since I attended the last time my event answers, and I would like to congratulate you on. I remember even 5 years ago I've been attending. I remember because of my localization, and I remember how it was. It's always quite like evening hours and so on, and topics we were discussing. So it's great what you are doing, my IVF Answers and the European Fertility Society, education, awareness, and the navigation for patients through the whole complicated and sometimes just, marketing, full of marketing and advertisement. And throughout all these rumors, you cannot find the exact factors or exact important things. So, and specifically the topic we are discussing today, this is it both very important and very discussable or controversial, I would say. Still, some people would call it like this. However, we have already our knowledge and the practice, which we would like to share about this topic and the point for the decision making process. We are never saying this is the only way to go. We are just saying this is an option to choose among the options, and then we are comparing the results which we expect to get based on the evidence we are having. So yeah, thank you for having me here.
Thank you so much indeed for your kind words, and thank you for introduction for this topic, as you mentioned. So as always, remember, we will start with Dr. Uliana's presentation. And for those who haven't had a chance to meet Dr. Uliana, she's the advisory clinic director at International Fertility Group, but also co-founder, director of clinical operations at Ovogene. And today we will talk about mitochondrial replacement therapies. She's a great person to discuss this. And of course, after the presentation, there will be time for your questions. So as always, please just type those in the chat so that Dr. Uliana can answer them for you. We will have around 30 minutes for the questions, so you can type those during the presentation or afterwards. Really looking forward to seeing your questions. And I think that we can go ahead with our presentation. Dr. Uliana will share all aspects of our topic right now. Thank you so much, and I think we can start, right?
Right. Thank you, Carolina. And again, thank you everyone for joining. I know for you it's a different time zones. For some of you, I know you've contacted me asking and checking about these details. So I really appreciate your involvement, and I appreciate about the fact that you are learning about the topic, that you are those who know already that you are supporting this technique, and we are moving. This is something what really motivates us to continue to learn deeper and to provide the services we can. But today I'm going to talk about the overview, so something what you already know or many of you already know, and the potential aspects of the development of this technology, what are the recent changes, and the opportunities around. So as the main player of this topic is the mitochondria. So let's talk a little bit about the mitochondria starting here. So this is a battery of the cell, and each plays the essential role in the generating energy for different types of the tissue in our body. And while we live our life, our body is selecting the most active systems and organs, which are called the central organs, and some others, especially while we are aging, they are receiving less energy. So this is a selective process, which is process of the aging and the development. And here it's a contrary to the process of the longevity, which can also be discussed throughout this topic, and specifically about the mitochondria. But if you are talking about fertility, so mitochondria is supplying an energy to the oocyte itself, and then to further to the embryo, which is developing using the environment of the oocyte. And this is very important to understand, that we will be talking today about the oocyte as a cell which has been generated or created in the ovary, which also changes with the different factors, if you're talking about factors of the age, or which has a genetic and non-genetic structures, which are cytoplasm and the nucleus and the mitochondria, and how they are communicating together. So if you're talking about mitochondrial DNA, we need to understand that as an oocyte has a structure which is called a spindle. So this is the nucleus, which has a genetic material inside. Also inside the cytoplasmic part, so in the environment which surrounds the nucleus in the oocyte, we have mitochondria. And mitochondria has its own circular DNA, which contains of 37 genes. They are encoding different RNAs, different proteins, and they are important for different activities as part of the electronic transport chain as well. So not all of those mitochondrial DNAs are causing mitochondrial disorders. This is also important to understand, because if we are talking about the technology itself, mitochondrial replacement therapies, they have an indication to be used for patients who are carrying mitochondrial disorder. And we will be talking during this evening about two different disorders. One, which is primary mitochondrial dysfunction, which is related to the genetic abnormalities for the patient, and secondary mitochondrial disorder, which is related to the aging process. But before we continue, it's important also to understand that the 99.9%, or we don't know this exactly, and in these topics, there will be a lot of points which we cannot answer right now. This is a point for the development and the learning process through the technique. However, we believe that the all mitochondrial activity, or maximum of this mitochondrial activity, is inherited from the mother, because sperm carries the limited mitochondrial DNA, which is around the tail, and there is a discussion either this mitochondrial is still active after the fertilization. But the mitochondrial DNA is coming from the maternal side. And how it's changed in the oocyte. So oogonias, these are very like the oocytes on the very, very first and early stage of the fetal development. They already have mitochondria, and their number is continuously increasing as the oocytes proceeds through the meiosis, and then reaching the potential to become a metaphase 2 oocyte. And the mature oocyte has some of the highest mitochondrial amount among the human cells. This amount is equal to the amount of the mitochondria in the brain or the liver, which are functioning very actively. So these great energy requirements, they are needed for the fertilization and the early embryonic development. And what is happening with the aging, so we will also discuss it further, so why we can fail in the process of the IVF treatment. And those oocytes which have low mitochondrial DNA activity, we receive them as the GV stage, or the immature stage oocytes, but those oocytes are not getting maturated while we do the stimulation. And also the numbers, of course, is increasing from the GV stage through the M1 to the M2 stage, if this is possible. So the primary mitochondrial diseases, these are those where patients carry the mutation and which is inherited through their mitochondria or the nuclear DNA. So also, it's not right to think that all mitochondrial diseases are coded through the mitochondrial DNA. So there is a part of the mitochondrial diseases which are coded in the mitochondrial DNA, and some others, they are still nuclear mutations. So we need to specifically understand, and there should be a genetic counseling performed for the patient who is a carrier of the mitochondrial disease. Either the mitochondrial replacement treatment is going to help such a patient to avoid the inheritance of this disease through that treatment. These diseases, they also are very tricky, let's say, because we cannot predict them. Sometimes they can manifest at any age. We've seen, I believe, who is following the news, we've seen an example of the prince who died recently because of the mitochondrial disorder. So once the baby was born, there was no sign of the mitochondrial disease. And then it manifested later. And unfortunately, using all the opportunities for the diagnostics and treatment, he died in the age of 22. So those diseases have no cure or effective treatment right now. And they are very diverse between the different organs and the systems. These are primary mitochondrial disease, which are genetically related. And there is a lot of foundations and the groups mostly funded by the families or the related organizations which are supporting and helping such groups of patients. And some of those organizations, they also supported and helped for the changes of the law, the legislations in the field of the IVF. For example, the Lilly Foundation, they were supporting a lot in the process of receiving the government review and the application about the mitochondrial replacement treatment being legal in the UK, which happened quite a while ago in the end of 2015. And as you can see, they are present in different countries. In Australia, there is a huge and very big organization in Canada and through the whole world. About very often patients are asking, because we are receiving a lot of inquiries from patients, different ages, different medical status, different anamnesis for all of those patients. And some of them, they are questioning the fact if they are carriers of the mitochondrial disorder. So maybe they can use this technology not because of the advanced maternal age, which is the dysfunction, like a secondary dysfunction, but because they are carrying this status. So it's important to understand that while we are in adult age, like over 30, most likely until that time, we already know if we are carriers of any kind of the disorder, but still there is a way how to analyze and how to check the fact about the presence of the mitochondrial disease. However, still this is not very easy. And currently there are different techniques, which is the genomics and the transcriptomics. So what is genomics? This is the way how to check the genes. And just quite recently, they developed a technique which is whole exome sequencing and the whole genome sequencing technologies. There are limited centers or the laboratories which are performing this testing, and the examples are below. So this is the Centogene, and they check for the rare disorders, and they check over 6,500 genes compared to the most frequently, like those which are offering most frequently the testing, which is between 500 to 600 as a maximum. And there is GeneDx, which do more than 20,000 genes and the hundreds of the rare diseases, both in the US and Oxford laboratory, which is also checking for the mitochondrial disorders. Then the transcriptomics, This is the way to analyze the RNA structure, not the DNA structure, but the RNA, which is also very helpful. And it can detect the mutations which are not detectable by the exome or the genome sequences. Metabolomics. So the metabolomic tool, this is to diagnose lactate and pyruvate, which are the biomarkers, because we are keen to, and also this is very frequent question from the patient, how you know the level of my mitochondrial activity, how you measure this, why are you saying that my mitochondria are not active? Maybe this is not the reason of the mitochondria, maybe there is something else here. So the answer now is we cannot do it right now, and this is a place to investigate, definitely, and we are open, and we already started some collaborations, scientific collaborations, about the way how to do it really properly, because the question for the patient, from the patient side, is not even about their own mitochondrial activity, because we expect it to be low, especially if we are talking about advanced maternal age patients. But the question is about donor testing, like how we ensure that the donor has a appropriate mitochondrial activity. And something what we can do now, we can check the efficiency from the previous rounds of the donation of a donor, and this is what we do. And we believe that this is a very important part of the how we can guarantee the best possible outcomes from this technology. However, we cannot specifically measure and say this is the level of the activities in the mitochondria which are here. However, these biomarkers have, like, there is a discussion about these biomarkers, and they have low sensitivity and specificity. So most likely we will be able to detect some other markers probably soon. From having the parallels with the recent changes in IVF, for example, less than 10 years ago, we had no IMH. We never measured IMH. We were focused only on FSH level, which is quite not sensitive, and now we are not measuring. And we have excellent results with patients who even are starting their stimulation with their FSH being over 30. So this is not a specific factor. Then other centers, they would say we cannot start the stimulation because here FSH is over 30. But this is not an individual factor. It should be a combination of the different factors. This is if we are having a parallel. So it's very possible that new markers will be developed soon, and we are happy to collaborate on that potential development. Functional genomics. This is the presence of the rare variants or variants of the unknown clinical significance. So those from you who have done already genetic testing, you can read that in the panel, maybe there was something detected, but it was said as a report from the genetic lab that this is clinically not significant, or it's unknown significance. So still in the human genome exome, there is a lot of those black parts yet. And this is, we need to understand that now we are talking about field which is not evaluated deeply yet, where we still have lots of question marks. As in IVF, for example, we can answer already a lot of questions except the implantation. So implantation, it's still something where we may say that still we don't have all answers here. We already can answer a lot, and we do this very successfully, but still there is a way to go. And the imaging technologies like the MRI, this is something just to prove or to confirm that we are on the right direction, assuming that we have this disease, but this is most likely to confirm the fact that it's already present. It's not to prevent or to, definitely it's far away from the genomics or the transcriptomics. So this is the way how to test if there is a question that we need to test this. However, as I mentioned already before, that with the recent advance in the genetic testing, mitochondrial dysfunction is being recognized to be more complex than we originally thought. And we are talking about also neurodegenerative disorders, metabolic disorders, and the cancer, where also scientists believe that mitochondria is playing crucial role. And there is already a suggestion how with the use of the mitochondrial activities or the transplantation of the mitochondria, we can solve, treat those disorders. But if we are talking about mitochondrial replacement therapy for in IVF, the primary mitochondrial disorders, these are those genetically inherited, and there are examples of those which can be evaluated or can be confirmed in the very early stages of the development. Some are later, but they are quite specific. And the secondary mitochondrial disorders, they are referring to the abnormal mitochondrial function. And here, the aging is playing very important role: inflammatory response, enzyme deficiency, metabolic changes, and all others. So this is something what is our biological clock is always running. Even now, we are going to spend the next hour or two. And during this time, we are producing our follicles, we are using our energy, we are losing our cells because of the process of the apoptosis. This is something what we cannot stop. That's why we need to focus about timing, which is so important. And this is something what we can measure, because something what we cannot measure, we cannot improve. We need to find a way how we will measure this. And after we are measuring, we are improving. But age, this is factor which we already can measure, but no one is guilty that something didn't happen before, because probably maybe we didn't know that something is possible. I'm talking about if we are discussing the preservation, like how to preserve the fertility function. There is a lot of educational activities about this, and I'm happy to support this as well, because I am a believer on the fact that if you can preserve something what you have right now, you need to do it immediately. And if you're talking about oocyte freezing, for example, this is something what you can do while you are producing your eggs, because there will be a process which is called menopause, and there is a statistics about the menopause. So you can see the map here. This is done by the like a huge society, so we can rely on those numbers. Even the colors are a bit different, but the numbers are not critically different. We are talking about difference in about 3 to 4 years, which is not significant. And the truth is that the menopause is delayed compared to the several centuries ago. Women, they were getting their menopause earlier, but also the lifetime of people in the world, that it was also much shorter. So where we are moving now, we are moving into the direction that the menopause is prolonged. This is great, and this is while we do nothing. So we are not talking about longevity programs specifically for the ovarian function. We are not talking about the ovarian rejuvenation or the stem cells or the exosomes or any other activation of the specific organ. We are talking just about the natural conception. But also, which is good. So it's great that the menopause is delayed. But also the average age of the first delivery in the world is increasing. And this is statistics from the UK. And it's very clear that in every 2 years, like in every 100 years, for the timing when the woman is delivering the first baby is increasing in 2 years. So if we will do these statistics for the future, quite short future, let's say 2100, so we expect women to deliver their babies in 51. Which solutions we are having for that? We are at IFG, we are treating patients, a lot of patients in advanced maternal age, and I did our statistics for previous year, and our average age was 42.8. And our average age starting from January this year until now is 46.4. So our population of patients is aging, or this is the population of patients who is looking for the solutions and who is looking for the solutions a bit beyond what was recommended before. Um, I can tell you that this is for sure more complicated population of the patients. And if I will get the statistics and we will share all this data with you, we have one more webinar scheduled, I don't know yet when, somewhere in June, I believe, where we are going to share the statistics, or we can do it separately, but it's not, we need a time to evaluate this from, this is as a cube, and we want to see it from all the parties. However, this is fact that we have patients, we have population of patients who would like to try the alternative opportunity for the oocyte donation. Because if you're talking about mitochondrial replacement therapies, this is something what already been said, that the only option is oocyte donation. So this technology is an alternative technology. However, I would be happy to work with the population of patients who are producing multiple number of the oocytes in the cycle. But it's not always the case, and we need to get better and to understand the things and to bring the solutions for those even who cannot produce multiple number of eggs during one cycle. And that's why we have something what is called cumulative cycles, and this is how we collect the desired number of the oocytes to proceed further using the technology. However, this is the way how we can be ready to shifting the first delivery age into these ages, and to be still successful there. So if you are talking about the ovary, this is the gonad, which serves as a source of the germ cells, which are becoming the oocytes, and there is a major production of the steroid sex hormones. So estrogen is very essential for our health status, for our cardiovascular system, for our mental health, for our like different systems and organs in our body. However, also the ovary is the first organ to age in the females, and this affecting both fertility and the overall health. So we used to say that this ovarian aging is a natural and inevitable physiological process, but the question, either it can be shifted or it can be prolonged, or how can we do it differently in order to understand what is going on in the advanced maternal age ovary, and which biological mechanism we can still make it work, like switch it on, in order to believe that this is possible to produce few more or some more oocytes. And this is also a part of the physiology of the longevity, if this is our goal. So depending what is the goal, either we are focusing on to solve the fertility issues, and we want to have baby in the shortest period of time, or we want to continue our hormonal activity and our activity and prolong our menopause. So if we are talking about oocyte aging, this is the progressive deterioration of the oocyte quality and the function. It occurs on the different levels. It's a molecular, cellular level, and also changes in the environment itself. It characterized with the changes in the cellular structures, genetic integrity, mitochondrial function, of course, as we mentioned already before, and also epigenetic changes. So we all know, I believe very well, the changes in the human germ cell number, that the majority of the cells, they are going through the process which is called the apoptotic arrest before the period of the puberty. And then we are having about 400 to 500,000 of the follicles which are potentially to ovulate, and out of those about 400 to 500 only These are the number of the oocytes which are produced as the mature or potentially mature oocytes, which are ovulating. All others, which are 99.9%, they are going through the process of the apoptosis as well. So we are losing the majority of our potential follicles if we are not freezing our eggs, if we are not even like that's very often it's a question about donating our eggs or freezing them for the social reasons for being used in the future. So this is something what definitely helps to preserve the quantity and the quality also of those oocytes which we are generating in the younger age. But if we are talking about this, of course, like no one said this 10 years ago or 15 years ago or some period of time ago. So that's why we have what we have, and we need to work effectively with what we have. So to sum up, while the organism age, we have the mitochondrial dysfunction in all the systems and all the organs, but there are more central organs which our body considers, and this is the natural conception, which considers to be the most important ones, and they still produce higher level of the mitochondrial activities, those cells. However, ovary is not the most important organ for our lifetime. It is, but for the short period of time, which we call the fertility period. But if we are already in the end of this fertility period, those changes are very obvious. So, because of the mitochondrial dysfunction, we have lower IVF success rates, we have lower fertilization, and higher aneuploidity rate. So the changes on a molecular level, on the cellular level, they were investigated by the scientists while they did the evaluation of the tissue of the ovary, which they received from 20-year-old patient and 40-year-old patient. So compared to the one who was 20-year-old patient, the tissue showed from the 40-year-old patient, the tissue showed reduced granulosa cell proportions, decreased proliferation of the granulosa cells, increased apoptosis in those granulosa and both granulosa and theca cells. And they are all, both these categories of the cells, they are important for the developing of the oocyte cumulus complex, because the follicle, this is the place which is growing inside the ovary. However, it can be an empty follicle, just a cavity, just a cyst, if there is no granulosa and the theca cells, because those cells, they need to be there in the proper concentration in order to generate and to help oocyte to be, let's say, born. Because if there is no oocyte cumulus complex formed, of course we will have no oocyte at all. And this is an explanation, like changes on the cellular level, changes on those follicles, and the fact that we just don't have simply these cells, they are not forming the oocytes. And this is the explanation why we are getting such an increased amount of the empty follicles compared to the younger population of the patients. And if we are talking about population of the patients who are 45 plus. Also decreased mitochondrial synthesis, ITF synthesis, decreased expression of the DNA repair and the mitochondrial function, increased oxidative stress. We can talk a lot about the oxidative stress, but of course, and you can tell this is because of the environment and so on, but some organs, for example, ovary, it's not an external organ. Like this is not the level of stress which our skin, for example, is receiving from the external environment. But it's still happening there. So this is something again about the inner regulations of the body. And also contradictorily, or just to compare, if we are talking about skin rejuvenation and the care we know and we do for this type of organ or system, we are very successful. And believe me, our patients are all young and beautiful. They look like a woman here on the left. Even they are over 40, because we know how to take care and we know how to rejuvenate and we know how to improve the functions or preserve the functions of this organ or the system. And we do nothing or almost nothing for our ovaries in order to preserve their function, to improve their function, and to make them work. So maybe the answer is here. Maybe if we can take more care about different organs, but again, it should be started some time ago, and it should be a regular approach, something what we don't have a culture to take care about our ovaries right now. We do not rejuvenate, we do not control, we do not prevent the aging of this organ. So maybe those ovaries would act totally differently if we would take care and use the rejuvenative approach. But also it's important to say that this rejuvenative approach should be local. It should be specifically for the organ, because if we can say like we are taking a lot of supplements, we do NIDs, we do different approaches, but remember that we have central organs which also need a supply of this energy and of all the activities which we bring with the supplements. But ovary is not considered to be the central organ, unfortunately, anymore in some period of time. So if we are talking about success rates, it's just an example, and this is an example about oocyte donation, but it's very similar with something what is going on in the technology of the mitochondrial replacement treatment, that we may, while we use the technique, but there are many milestones, of course. The first milestone is to produce the oocytes. So for many patients of ours, we are not talking about the technology itself until we are successful in accumulating the number of the oocytes we wish to have or we think will be reasonable to proceed with for using the technique further if we are talking about advanced maternal age patients. But the first aim is to get those oocytes. And then the second aim is to create blastocysts. Here we have different players already, and the big part of the responsibility is from the donor oocyte, and this is very specific selection. I will talk about this a bit further. And then the third milestone is the euploid embryo formation. So we strictly recommend still to test the embryos which we are creating through this technology. And then the fourth one is an implantation potential. And then again, of course, the live birth. So this is like the 5 main milestones. But if we are successful in the first milestone and we have the number of the oocytes we expect to have, and quite often, and these are the medical indications for the MRT, these are patients who have several repeated IVF cycles with either no blastocysts generated, even if they have an excellent number of the oocytes. And it's happening that we are getting over 10 oocytes in the cycle for the patients over 45. So there is a specific category of those patients. However, still the cellular level changes, they are same. And the percentage in terms of the potential of the euploid blastocyst formation will be the same for all. Like if we are talking about potential 10% of euploid blastocysts, we need to have 10 blastocysts in order to get 1 euploid. We are not there. We are not in that numbers. But if we have a euploid embryo formed, healthy embryo, the implantation potential and overall success rate, also including the uterine factors, of course, but taking into account that the uterine factors are also normal, we will be there in a potential of 50 to 60% clinical pregnancy rates for any age range of the patients. But if we are using our own IVF, like we are going through the whole process, we created our own oocytes, and then we just did a regular IVF cycle, then you can see how the percentage is dropping starting already after 35. And this is the statistics which I believe you all know. So this technology is giving a hope and the opportunity to be in that line, horizontal line in between 50 to 60%. Of course, we can always evaluate individual potential, and this individual potential would come mostly from the implantation potential, but not from the age or other factors. So this is exactly the statistics which is showing the rate of the genetic abnormalities and how they increase with age. And you can see that there is no correlation with the number of the blastocyst patient is able to produce or the number of the blastocysts which were evaluated. And even for the oocyte donors, which are, we expect them to be young and healthy individuals, which they are. However, on the cellular level, still we have a genetic abnormalities. We all know very well that not every oocyte is a mature oocyte, and not every oocyte will be having a healthy genetic composition. So statistics shows that about 30% of the embryos generated from the oocyte donors will be unhealthy. And then how it increases with the age. And this is almost like every 2 years increase. So there is no statistics here for 45, 48 and over. So, but of course, the statistics is increasing. So this shows that in order to be successful in the age over 40, we need to have really great numbers of the oocytes and great numbers of the blastocysts. And this is very challenging. So to prove the data about the genetic abnormalities on the level of the oocytes for young and healthy population, we've done at Ovogene the study, which shows that in average 25% of the oocytes from the young and healthy individuals who are oocyte donors, and they all were between 21 to 30. Like here, we didn't have patients who were older than that age. They are coming back as genetically aneuploid on a stage of the oocytes. We haven't tested advanced maternal age oocytes. Like this data, we started to test it, but this data is still very, very limited. And we should either do this testing for the scientific purposes, because if we do it both for the clinical purposes and the scientific purposes, we are just using those oocytes, and we will not be able to use it further for the clinical like for the treatment itself. So slowly, but we are building this data, and I will be happy to share it with you, because this can be one of the theories why and how MST or PNT is working. That it's not obviously that all genetic information and all the nucleus from advanced maternal age oocytes on the initial stage when we are receiving the oocyte, they are all having genetic abnormalities, but they are all getting as an aneuploid embryo. So why? What is the impact and from where? So the theory is saying that impact is coming from the environmental changes. So as we said in the beginning, that the environment, everything what is going on in the cytoplasmic part of the cell, this all is already changed. We don't have the proper mitochondrial activity. We don't have proper intracellular communication, metabolic activities are different, biochemical changes, and so on and so far. So this can be an impact of the environment. If we are talking about rejuvenation, or if we are talking about just a complex solution for the continuous stimulations or the cumulative cycles, how we call them. So we want to drop the number of early primordial follicles. Like this is something what we want to solve, to increase the number of the primordial follicles, specifically through the constant monitoring, because it's quite often happening that if we do a monitoring once a quarter or once a month, we are not seeing all the primordial follicles which are potentially to be stimulated, because there is a follicular wave. And this is the theory which already been proven, that the follicles are growing not only in the follicular phase. We used to start stimulation, all the stimulations we started starting from 47 years ago until recent time on day 2. And there were a lot of publications that day 2 is better than day 3, definitely better than the day 4 and 5, and there will be different outcomes we will be receiving. But we never talked about luteal phase stimulation or so-called random start, and specifically for advanced maternal age patients, where sporadically just single oocytes are showing up, are growing. We just need not to lose them in that specific period of time, and just keep them involved, feed them with the gonadotropins, and by this way they can be stimulated. But if we don't do the monitoring, how we could know that they are there? They are silent. They are not producing enough estrogens, so we will see it as the ovulation. It will be able We will be able to control this only through the precise monitoring. Uh, we want to solve the issue of the increased follicular destruction. So we believe that here something what will be effective. We still need more data about this, but the preliminary data, and also most of the data is coming from the Asia, from Japan, Singapore, China, Korea. We have very limited data here in Europe because of the regulations and also the conservative approach. Like, we even don't do PRP as much as we could. Like, we are not talking about the stem cells and the exosomes and anything about the rejuvenation almost at all. So this is all the opportunities. We want to decrease the number of the apoptotic follicles. So apoptotic follicles, you learned already, those which they don't have the theca and the granulosa cell. They don't have normal cells which need to support the follicular development, and but not only the follicular development itself, but also the oocyte cumulus complex formation. And also the inability of the follicles to respond to the gonadotropin stimulation. So we know that there is a stage when the follicles are gonadotropin non-sensitive, and this is mainly very early stage of the development of the follicles, and then they reach to 2 to 4 millimeters. They should become gonadotropin sensitive, and this sensibility is coming also from the cellular configuration of those follicles, and if they are viable, they will be responding. If they are not viable, they can show up, they can grow until 7, 8, 9 millimeters, and then they just will disappear, meaning this follicle was not viable. There was just nothing. It's gone. So this is all physiological changes. So what we are going to talk, that was like a preparation, and now we will talk more about the technology itself. So we are going to talk about oocyte and the zygote reconstruction. So from those who see the screen, again, we can see the cell, which is a very big cell in our body, which is oocyte, and inner structure is a spindle, and there is a big cytoplasmic volume around it, and there is a membrane on top of this. And on the right, it's a zygote. So we have two pronuclei. These are two genetic materials. One is coming from the mother, and one is coming from the father. And also they are surrounded by the cytoplasm and covered with the membrane. So while we are creating an oocyte or a zygote after the mitochondrial replacement treatment, which has two different techniques, one is called MST, spindle transfer, maternal spindle transfer, and second is called PNT, pronuclear transfer. We are transferring those genetic materials, only the inner structures, from the cell where they are into a new cell, into a donor cell with the totally different environment, young and healthy environment. Either this is on the oocyte stage, which is day 0, immediately after receiving the oocytes, or on day 1, which is not completed day 1, but between 14 to 16 hours post-fertilization. And I will show further how it works. But before we will continue, because we are reconstructing the oocyte, so this is what we are doing. We are not doing cloning. We are not doing gene editing. We are not doing anything else. We just reconstruct the cell. We are taking part of the cell from the environment which is not optimal already because of the changes or because of the genetic predisposition, and we are transferring those parts which are essential for the further development into the new cell, donor selected cell. And as I mentioned before, we cannot measure the mitochondrial activity, not yet, but I believe that very soon in the future that will be possible. But quality of the donor oocytes is very important for the successful of the mitochondrial donation. I believe you know that there are different groups and the different scientists and the doctors and the embryologists are working on this technology. There is no uniqueness on that field yet. The protocols are not shared. The results are very much different from one facility to another. Some facilities, they have exclusion criteria which are related to age, for example, many of them. So this is something what is the limiting factors. Other facilities, they also, like more and more groups, they are starting clinical trials, and this is great. If we would be able to do a clinical trial and the funded process for this treatment, we would just we know already, because of communication with different women and many patients, we know that there is certainly a group of patients who would be happy to join the clinical trials, but so far we don't have one. But we will be very open to start or to collaborate on this matter, as well as we are working to have a collaborations with different funds who potentially can support this technology, and through these collaborations we will be able to support our patients as well. But so far this is not the case, but for sure we are already like putting our responsibility on that part as well. So the quality of the donor oocytes, it's very important. So big part of the success of this treatment will rely on properly selected and really selected donor oocytes. Not every donor is a best donor. We know this very well, believe me, because there can be a young, healthy, fit and slim, and who's following the healthy lifestyle, and even she has her kid as having a proven fertility, and the oocytes are looking beautifully as through the morphological selection, but such a donor will generate 30% of the blastocyst. And there will be another donor, maybe a little bit older, having a kid or having no kids. Their oocytes will not look morphologically as AA, maybe as AB, but they will bring us 90% of the blastocysts. So this is all about the potential of the donor, and this is all about the proven fertility. So for this treatment, we include donors strictly, those who have her own child, because as we spoke on the beginning, that the inherited mitochondrial disorders, we will see it while the baby is born, if there is something there. So this is like a one factor to be proven. Excellent previous efficiency results, both in the morphological characteristics of the oocytes and the morphological characteristics of the blastocysts, because we need to work with those cells. This is a very precise micromanipulations, and this is very important to have a proper formed cell and of the cell of the best quality. And also the generated blastocyst rate and the euploid blastocyst rate and the previous clinical outcomes, having a clinical pregnancy using the oocytes from this donor, and those oocytes should be strictly fresh. We have nothing against vitrification. We believe that this technology works close to 100%, and this is definitely to be for the oocyte banking and for the oocyte donation treatment. But for this specific process, also because of the micromanipulations, those oocytes needs to be fresh. So this is our implemented AI algorithm as well, like how we select the oocytes. We also do this for our patient oocytes. So this is how we define either the MST or the PNT technique will be preferable for this specific patient. Part of this information, either that should be PNT or MST, we are getting from the medical records of the patient. So we always evaluate all previous outcomes And we never, or almost, we don't have patients who had no experience in IVF. So all those patients, they had repeated stimulations, they had no blastocyst or no euploid blastocyst in the past, but we can learn a lot from their embryological records and the cycle outcomes. But also in a time when we receive the oocytes, we do this morphological selection and evaluate the clinical information also from the donor and the patient. This is how we can predict the further step, and we know which technique will be the most beneficial for the patient. So this is just for those who visually receive the information better. The difference between the oocyte donation and the mitochondrial replacement treatment. If we are talking about oocyte donation, so baby will be 50% genetically related to a husband, to your partner, but there will be no genetic information inherited from yourself, even if you are carrying a baby. We are not talking here about these epigenetic factors, which are so this is not a genetics. This is something epi, but no genetic DNA. And if we are talking about mitochondrial replacement therapy, the baby will be genetically related to both of the parents, to the mother and the father, to the mother through the nuclear DNA and to the father through the nuclear DNA, but which is, as we said, that the mitochondrial activity is not there after the fertilization. So it's 50% to father, and we cannot precisely measure right now, but we, from everything what we learned through the process, this is 99% and more to the mother, like 49. So another theory, so I was sharing with you that the theory is that we don't know how many mutations, and if the oocyte, which is advanced maternal oocyte, is carrying this genetic disorder on the beginning of the process. So just once the oocyte was collected, and if the environment is forcing these disorders to happen, this is one of the theories. And another theory is the theory about the self-correctness in embryos. So there are publications about the fact that even if the oocyte or the sperm has a small disorders, which we believe that those which are calling to the mosaic embryos in a final outcome of the development of the embryo when we do the PGTA testing. For those who already did this, you know that there can be a 3 category of the embryos which we are receiving post-testing. Genetically healthy, those which are called euploid, genetically not healthy, aneuploid, and those which are mosaics. So the fact is that after this technology, which is either MST or PNT, we almost do not receive mosaic embryos. And we will also be happy to share the statistics. We already prepared the data for the publication, but we want to publish this in the high impact factor journals, which is taking a lot time for them to accept and to review. However, I believe it will happen very soon. So the mitochondrial replacement therapy, this is very important to remember that we cannot change the nuclear DNA status. So if those nucleus was carrying already in the beginning of the process genetic error, we will not be able to correct this if this is a significant genetic error. If this is non-significant, yes, the mechanism of the self-correctness most likely can work. And here we see that after we have a zygote and then we have the DNA repair mechanism, which depends from the different and the complex factors, after these factors impacted, there is a two-way. Either the apoptosis, meaning that the embryo is stopping its development and just dying, which is happening often, and I believe you've seen this, that embryo developed until day 2 and then on day 3 it stopped, or especially a lot of the embryos for advanced maternal age between day 3 to day 5, probably because of the lack of the energy and all the different functioning changes, or it repairs. So the self-correctness, this is the second mechanism, which is what is helping to self-correct, this is the cytoplasm of the donor oocyte, which is young and healthy environment. So the technology itself. This is how we see the spindle. So not every laboratory can evaluate a spindle because there should be a specific equipment which is called polarized microscope. With the polarized lights, we are able, if we configure the oocyte properly, we are able to detect the spindle. So this is also a way that you need to know how to search for this, and you need to have an equipment to search for this. And not every oocyte is having a visible spindle. So, and also the characteristics of the spindle can be very different. So this is one of the approaches how and why we can recommend one or another technique, but this can happen only after we received oocytes in the laboratory. And what is happening after, when we detected the spindle, if we decided that MST is going to be a technique which will be performed for the specific patient, the spindle will be removed from the maternal oocyte and transferred to the donor oocyte. Before the spindle is transferred to the donor oocyte, from the donor oocyte, spindle is removed and destroyed. So this is the oocyte reconstruction. After the spindle was replaced, transferred from the maternal oocyte to the donor oocyte, fertilization is performed. And the cytoplasmic part of the oocyte from the mother is destroyed as well. So here, Carolina will help me to play a video for you, so you will be able to see how it's happening, that this is the oocyte, what you can see here. And now we switched on the polarized lights. So it's very dark now. So the oocyte is clearly seen. And then you see the white spot. This is the spindle, and the needle is going inside the oocyte. And the embryologist is aspirating the spindle very gently and keeps it in the needle. This is the maternal oocyte. So you see a very small hole. This is the process which is called fusion. This is also a very important process, that we are able to make sure that there is no cells of the cytoplasmic environment around the genetic material. So it should be well cleaned spindle. And then after it was cleaned, it transfers back to another oocyte. You see that this is a different oocyte. There was no hole on this oocyte. And then the spindle is inside the new oocyte. And then after that transfer, the fertilization will be performed as a regular ICSI process. And the ICSI process, this is the one which we usually or always do. So what is very important, that the spindle complex, because it's spindle chromosome complex, is sensitive to the micromanipulation, and only specifically if there is a medical indication to perform this type of the technique, we would choose to perform these techniques because of the sensitivity of the process. There is something what is needs to be, we need to know how to do this. This is the visualization of the spindle. And of course, this technique is very much operator dependent. And the different technique, this is the pronuclear transfer. So on day 0, when we received the oocyte, we fertilize both sets of the oocyte because they need to be synchronized. If they are not synchronized, so the donor oocytes will be fresh, but the patient oocytes will be frozen. And we are experts in the vitrification, and the material is surviving 100% if it was frozen by us. There can be some different statistics if material was sent from overseas, from different clinics, because obviously that we are right now we are receiving materials from different clinics from abroad, and we are happy to collaborate this way because this is very helpful for the patients not to fly, because this is a medical tourism. Obviously, because of the regulations, it's not easy to be performed in home country of the patients, but that's fine. And we always discuss with the clinic how to vitrify the biological materials and how to prepare them and send them over. So the pronuclear transfer, either we create a zygote in our facility or we receive the fertilized zygote. Then the day before, we will do the egg retrieval for the donor and create the donor zygote. And then day one, post egg retrieval, the whole transfer will be happening. So this is very similar, and let's play the video here so you will understand. Just we don't need specific lights because we can already see both pronucleuses, and as I said, they are coming one from the female and one from the male. So they are both here and very gentle as well. You can see that this is a different diameter of the pipette, and the pipette is a different shape. So this is also like this technique technically is a bit more easier because the structures are formed already. They are easy to manipulate. So both are here, they are in the pipette, and they will be transferred to the different oocyte, which is a donor oocyte, using the same way as we previously just watched as a transfer or the reconstruction of the oocyte. Thank you so much for playing this. So it is important that once we created embryos, after we did the reconstruction, and obviously after this technology, embryos are developing from 5 to 6 days, which is a normal development, the term for the development of the blastocyst. They usually are developing in time, so, and it's normal time. Most of them will develop on day 5, but some will develop beginning like very early day 6, and there is no difference also about their performance and the implantation rates. And then after they are created, they need to be tested. We strongly recommend that all embryos will be tested because we, using this technique, we cannot avoid complex aneuploidies. And this technique is not a magical technique which will solve all the genetic issues. No, it will help to increase the potential of euploid blastocyst formation for each individual case. And we have certain statistics about the euploid blastocyst formation, but this is not a guarantee for each individual, and we cannot avoid those structural chromosomal disorders like the trisomias and others. So that's why this is very important. This, again, this is not gene editing or something that we are cutting those genes which should not be there. We are just helping to preserve the most potential for your nuclear DNA to develop in the best conditions possible. A little bit about the regulations globally, because this is very sensitive topic and patients are asking very frequently. So the first mitochondrial donation regulations was accepted in the UK in 2015 after huge discussions and after the clinical case, which was a woman who forced or who helped these regulations to come in force, also with the support of different organizations, as the patient organizations, as Lily, for example, as I mentioned before. So this is a great achievement. However, individual clinics, they need to have a license to perform this treatment. And so far, we do not see a huge amount of cases happening in the UK, either because of the way how to detect the mitochondrial disorders, because that is not for the age-related changes. It's not for the secondary mitochondrial dysfunction. It's for the primary mitochondrial diseases, which needs to be proven, or maybe something changed, like the staff or equipment or the technology from the perspective of the clinics which were providing this treatment. But that would be great if they continue those treatments, of course. And the mitochondrial donation law, MEVS law, which started in, was voted into 2022 in Australia, and there is a huge support so far on the level of the clinical trials yet. So there were no clinical cases performed even since that time until now. But it looks like it's a very active position of the society and also the policy members and the professionals. So we believe that very soon Australia will also have very first cases. So few publications which we were able to to make, because why I'm saying, because we need, it's a lot of work to generate and to gather all data together on the way and such a sensitive topic, and then to went through the evaluation committee and the decision making for the publications. But we know this is our responsibility, and we are very happy to do this. And I believe very soon you will get more publications, meaning that the data is proven, and this shows the success of this technique. Because I personally, I believe in this technique. I believe on the fact, as I mentioned before, that I believe that we need to preserve what we can preserve. And this is the reason why I froze my eggs in 41 last year, and I hope to freeze one more, to do one more cycle now in 42, because you know, I am sometimes very motivated from our patients, which are many of them, they are much older than that age, and how brave they are on this family planning and the family building. So once kids are already almost teenagers, this is always a room to think about. So in terms of the ethical issues, it passed already the ethical review from the Nuffield Council of the Bioethics. It's about the regulations in the UK. So it was also a lot of discussions, a lot of yes and no, and different positions. But finally, there are some open questions, of course, about the maternity rights, about the donor anonymity on this field, and so on, and the long-term health evaluation of those babies, because obviously now the oldest baby was born in 2016. So if we are taking the long-term risks, so we cannot answer those questions. But if we, for me, it's always correlation with the IVF itself. It's less than 50 years when we are practicing, and we have millions of babies born all over the globe. And if we would not, there would be no brave people to try this and to implement this, there would no solution for such many, many, many individuals globally. So this is what we believe. And in terms of the future developments about the mitochondrial transfers, they are going beyond the IVF. And they are going to a discussion that we need to restore the mitochondrial function in the diseased cells. And specifically, we are talking about those age-associated disorders as the neuroendocrine and the neurodegenerative diseases like the Alzheimer and sclerosis, but also the metabolic disorders and even a cancer. So these are all just very briefly. I did this today, in all published in 2025 already, and as a solution for different disorders, as you can see, different therapeutic approaches, cancer, and different other issues. So this is where we can potentially develop, and definitely we need a collaboration between the professionals in the field. Like there is a lot of legal side, ethical side, different professions need to take care of this and evaluate the risks, and so on. But I believe that we will be talking about this also very soon. So far, I'm sorry for taking that long. We are ending our presentation, and I would like to thank our IFG team. And this is the collaboration of the expertise and the extraordinary care. We have embryologists, innovative and just unique ones, IVF specialists, reproductive immunologists, geneticists, nutritionists, endocrinologists, psychologists, IVF coordinators, and the care team. So all these people are doing possible something what it was said quite often before that was impossible. So I'm really very motivated from all the results we are getting together. And I would like to thank you, those who trust us, and we have results together. To conclude, mitochondrial transfer technique, this is the treatment which prevents inheritance of the mitochondrial genetic disorders, but also can serve as a solution for the secondary mitochondrial dysfunction. Most effective techniques are the mitochondrial transfer and the pronuclear transfer. I was not even mentioning the cytoplasmic transfer, which it's not working, and there is no discussion about this. Successful mitochondrial replacement therapy needs a proper laboratory protocols and the excellent micromanipulation practices. They are not same in the different locations and the different clinics and in the different groups, believe me. Blastocysts which are generated through this technology, they need to be tested. This is safety of this process, as we cannot avoid the genetic disorders. We are just helping your potential DNA, which is healthy in the beginning of the process, to grow in the most optimal conditions. The selection of donor oocytes is crucial for the success of the MRTs. Ongoing research and the establishment of the standardized protocol are essential. Research are already here, and the standardization, I believe this is a bit longer goal to achieve, but still it's very important. And the education and awareness for patients are vital in helping patients to make informative decisions. So we are committed to excellence through the personalized and the expert approach, innovative mindset, and the compassionate care. You can contact us at IFG, and we always will be able to provide the most detailed information and share all our knowledge and practices. I was not touching statistics here, but we are going to have another meeting for this. I know you have a lot of questions, but what I would like to say about this, that it's very not right to compare results, because we never had 10 individuals which are the same, even if they have the same age, very similar IMH, maybe quite similar antral follicle number, but these are still very individualized response in all treatments, also for the embryo transfers and so on and so far. So again, thank you for your involvement. Thank you for your time. Thank you for listening, for all your attention, for all your questions. So I see they are already here. So let's move to the question and answer session.
Amazing. Thank you so much, Dr. Uliana, for walking us through all aspects. It's not an easy topic, but I think that it's both very useful for you and very interesting indeed. Lots of details to remember after this as well, but thank you so much. And you are very right. Lots of questions are right here as well. So I will go through all of those questions, of course, but remember that that the time is limited. So if we are not able to answer all of them, please remember you can get in touch with Dr. Liana and her team at IFG directly, and I'm sure they will be able to help you out. Most of the time, you will also need some more personalized information. As Dr. Uliana mentioned, every patient is different, so please remember that. But of course, right now we would go ahead with the questions. And I think that this question, I will not go for the first one, we will go back to this, but basically, I think it's one of the questions that will come a lot, right? How safe is this? I mean, are all the babies born normal? If you can tell us a bit, we already discussed this, how many babies were born, if you can tell us a bit on this as well.
Yes, sure. This is the most common question, I believe, as well. And I totally understand and support the fact that we need to try to answer this question. Frankly speaking, I am not sure that I will be able to answer it fully, because as I mentioned, that the first baby which we know was born using this technique, it was in 2016. Our first baby in our group, which treatment was performed in Ukraine, was born in 2017. And since that time, we have 29 babies born in our group. So we are in touch with majority of the parents, not with all the parents. Some they were not happy to continue. This is very voluntary process, I would say, but majority of the patients, they are providing information about their health status of their babies. We created the newborn assessment forms and we are collecting them. However, the oldest baby is still 11 now. So we need to understand that this is not the long term perspective. And we need to take this into consideration and as a responsibility. But so far, there is no physical or mental or any other type of the development is not different for those babies who are born specifically in our group. And you need to know that I think that this is a bit underreported. So I personally believe that there are more babies globally, just not every team who is working on that would share these statistics.
Okay, crystal clear. Thank you so much indeed. And actually, there is a follow-up, so let me go straight to this one. Are you able to tell us this? Out of how many patients total did those 29 births come from?
So the total number of transfers was over 70, but we need to understand that when we calculated those 29, we still have many ongoing pregnancies. So we were not calculating this as a separate because this is for the period. And for the period, we have like latest statistics as 56% of the clinical pregnancies for all the population. And our population is between 39 to 52 years old, those who are accumulating their eggs, and those who, the oldest woman who gave birth, she was 48. So those between 48 to 52, we don't have the live births yet. But it's still possible, and this is very individually. If you would tell me even 5 years ago, because I'm consulting patients for since 2008, and majority of my patients, they were oocyte donation patients because of my dedication to the third party reproduction. So when I learned about this technology, I truly believe now that patients need to know about this opportunity and just select the path they would like to proceed with. Meaning, one way is just to continuously doing IVFs with obviously very low potential to succeed. And this is emotional burden, financial, and waste of time. As for me, oocyte donation is an option, but we just discussed what is a difference there. And the mitochondrial replacement therapy, it's an innovative approach which merges both your own IVF and the fact that you are having genetically related baby. But the only really, like, frankly speaking, question, this is the long-term issues, but through the technique and the technology and the process, how we know it is performed, we do not expect any potential risks. As we didn't expect them. Like the most common question was either the Louisa Brown is going to be fertile, if she's going to be able to have kids on her own. So we already have this answer, but we need a bit more time.
Of course, that's always the case, I'm afraid, right? So yeah, we need time. Okay, thank you so much. There are a few questions that are very similar, and I'm sure you are getting them again quite a lot. Those are personal questions. Simply, there's a question about I'm 43, 44, 44. So could I use this technique to conceive? I have menopause, right? So this is one of the questions. I will show you a few of those because they are very, similar. Here we have a patient who is 50. Is this technique possible for her? So as you can see, many different ages, and of course AMH here as well is mentioned, and they are quite similar. Anything you can advise?
Yes, so we are happy to evaluate each and every situation separately, but something what is very important to say here, that age is important, but this is not the most important factor. Something else what is important, so ideally we would evaluate this all in one. So age, the fact if patient is still cycling, so if she is still cycling, this is a positive predictable parameter. If she is not cycling, then we need to understand how long ago she stopped to cycle. And then after we have this, we would need, of course, the hormones. We need IMH level and the dynamics of the IMH level. That would be ideally. So, meaning what is the IMH today and what was your IMH like how long ago before you measured this? If you measured last month, share this with us. If that was 3 months ago or half a year ago, share the previous IMH. So we would understand the decline in which, let's say, stage of the potential premenopause or the menopause you are. But if you are already in the menopause, then we would evaluate also how long ago was the cycle, what's going on in the hormonal level, on the hypophyseal level. And then, so IMH, FSH as well, and the dynamics of the FSH, it will be helpful. And then the antral follicle number or the just pelvic scan details, taking into account the volume of the ovaries, not only to measure if the follicles are there, but also the structure and the volume of the ovaries, and of course, if the follicles are there or not. So all these parameters, this will be something what we would need to evaluate before we will provide an individual answer for each and every individual patient, because it can be very, very different. Like, as I said, I would be happy to treat only patients who are cycling regularly, who have relatively high or reasonable IMH, but it's not always the case. And here the collaboration is important. It's important to understand the fact and to accept the fact that we are working with very last potential follicles which are possible to reach and to get them maturated and to receive the viable eggs. But we need time. We need to be flexible for this. We need to maybe even believe in yourself and trust to the technology and to the doctor, and then just to continue moving, because specifically for those patients we will have also unsuccessful cases, and here there will be a question when to stop, and definitely we will tell that we would recommend to stop. So we would also advise this to the patient, and then it's not the end of the story. There is always an oocyte donation as an option. As for me, this technology is something just before the oocyte donation, just to give the last trial for your body to produce the eggs, embryos, healthy embryos, and help them to implant. Implantation potential is a different question. So we are very specific on how we prepare patients for the embryo transfer, because this is important and this is our statistics and the statistics of the technology. So we want you to be successful in your embryo transfer technique, which is almost the final stage. However, sometimes patients are insisting, even those who have terrible uterine conditions. But if they are terrible, as well, we need to evaluate. Sometimes they can be improved. So this is as well, again, very individual. So I cannot answer each question here, but if you will share your medical history, having all these details in there, we will definitely be happy to evaluate.
Perfect. Thank you so much. That's why I wanted you to see there are plenty of questions, of course, that require simply thorough evaluation. Yeah, so of course, thanks for that. And remember, you can get in touch with Dr. Uliana and her team. Okay, next question that we've got is here. Are you using only mature M1 or M2 oocytes for MRT, or are you exploring use of germinal vesicle oocytes?
So in our protocol, we are using mature oocytes for the technology. So we are starting the process once we have mature oocyte or the already created zygote. If we are getting M1s or GV oocytes, we are maturating them. So the process will continue after they reach to the mature stage of the development. We know that there is a group who is suggesting that you specifically need to retrieve GV stage oocytes. We are not in direct communication with them, so we cannot ask questions we want to ask, unfortunately. Specifically, how we see and how we see the difference and how we evaluate as well potential risks through the transfer on the stage of the germinal vesicle. But we are not saying, like, because we cannot evaluate, we are saying this is our protocol and this is our technique, which we know how it works. If that group will be able to publish their data and we will be able to discuss or evaluate, it will be just great. So maybe this is also something what works, but I cannot comment. I have no information to comment on that.
Sure. All right, thank you again for that. How many oocytes do you need approximately per cycle? This is the question.
So we had different cases, and we, based on our statistical results, we believe that starting with 4 zygotes, like not 4 oocytes, but 4 zygotes, this is something very optimal, where we already expect to receive at least 1 diploid blastocyst. And for the technique in general, we include 15 oocytes as maximum. Like, this is our current, let's say, approach. But for many patients, we don't really need 15, because this is too hard to accumulate that number for some specific patients. If that's possible, of course, it's a choice of the patient, but it will be a good number to start with 4. Also, we had cases with starting with 1 egg and healthy embryo, euploid embryo, successfully implanted, and the lady is pregnant now in the 3rd trimester. But this is more like a clinical trial, clinical cases, not the statistics. I remember also case where we couldn't reach more than 2. We had 2 eggs, 1 euploid transferred, not successful. But patients decided also in parallel with the MST or PNT, they did also egg donation. So they fertilized 4 eggs from the donor, and we received also 2 euploid blastocysts from the donor. And she is now pregnant with the donor embryo. So she tried. It was not an option. It unfortunately didn't work with the first embryo transfer, but she is still successfully pregnant. So here is about acceptance of the next steps, let's say.
Mm-hmm. Okay, and if you can take a look at this, which is has a highest success rate of euploid embryo and of live birth, MST or PNT?
So majority of the statistical data and something what we do, like majority of cases are PNT cases, just because of the accumulation of the zygotes, and quite often they are frozen somewhere else, not in our facility, and then sent over. So zygote is more stable structure. So majority of the cases would be PNT cases, but from those MST cases, which were MST, statistics is not significantly different. So we can say that there is no statistically significant difference between MST and the PNT.
Mm-hmm. Okay, just clear. Again, thank you so much. Is there a waiting list for that procedure? This is the question that we have.
No, there is no waiting list, because we understand that we are working with advanced maternal age patients where we never suggest to wait. So if you want to start stimulation, you can start it when the conditions are for this. We are happy to evaluate your stimulation if you are starting this at home, or if you want to be supervised by our team, you can always just come to Tirana, and there is a clinic which can monitor you there. It's busy, but it's not crazy busy. So we always accepting new patients and at any time. So there is no waiting list, of course.
Okay, thank you for the reassurance here. All right, the next question. What is the recommended trigger size and freezing protocol to bank eggs for this?
Thank you for this question, but again, it's very individual question, because we are having many accumulated cycles, as I said, with our patients. And for many patients who are starting with us, it's not the first cycle. So there is already some learning point from their previous stimulation outcomes. And of course, we would to just simply answer your question, I would say it's between 17 to 20. This is when we would trigger the patient, but it will also depend on very, very different factors, like what would be the estradiol level, during how long this follicle reached this size. Either that was a very rapid development, which is more than 3 millimeters per day, or it was very slow development, which is less than 1 millimeter per day. So the decision point here will be very different. And what was the previous outcomes based on the longitude of the stimulation, type of the trigger, time, how long this follicle was developing for, level of the estradiol. If we can get this, we of course will evaluate and then will make an individual decision for this specific patient on when to trigger.
Okay, thank you. Again, as you can see, lots of other questions, detailed ones as well. But let me just show you this one. You mentioned this before during your presentation, in fact, but what are the current technical barriers preventing the wider adoption of MST and PNT?
So if you're talking about technical barriers, because as I would say, they are not technical, it's a global regulation and the acceptance of the innovation. So I would not call these technical. It's more like a global perspective. But if specifically a question is about technical, it's just the people. Unfortunately or fortunately, it's a very operator dependent technique, and not every embryologist, like the embryologist, has a skills, and this is something what either we can do excellently those micromanipulations or we cannot. So I believe that this is very personal knowledge or the practice which not everyone is given. And the another barrier is in the protocol. So the protocol, actually one protocol was published, and I know that many embryologists are trying to follow this protocol, but it doesn't work well. So there is an adaptation of the protocol in order to achieve the best results, and then the groups are not sharing that protocol because of there is no general policy for this technique, I think. So that's why these are probably the technical limitations. Equipment, but this is something what can be solved, of course, but this is quite specific as well.
Yeah. Okay. Thank you so much. There's a follow-up in regards to the previous question as well. So you mentioned 29 births out of 70 transfers. Was that 70 patients, or did some patients have multiple transfers? I'm curious how many patients had birth out of total patients.
So this is from per transfer, not per patient. And yes, we had several patients who had multiple births. We have two, and the third family, two already families which have multiple, like twins, and one more family will have also a new child very soon. So 3 patients out of all these numbers, they were repetitive transfers, but repetitive successful, if we are talking about successful. Also, we've had repetitive unsuccessful, of course, in that group.
Okay, thank you. I'm just writing a message, but thank you so much for that question there. We still have a few minutes left. We will be slowly finishing. As you can imagine, we could go on for hours when it comes to this specific topic. But let me just have a look. So this is the question. Can you select a donor who looks similar to you so that if the PNT fails, you can use the donor embryo and feel okay about it?
So usually if patient accepts the fact that the donor embryos needs to be selected, we are giving an option to choose a donor or share with the patient the phenotypical criteria of the donors who are going to be used in this technique. We are matching donors with the blood groups, so this is something what still will be matched, but we do not match them with the phenotypical characteristics if we know that the oocyte donation is not going to be a treatment option here because the phenotypical characteristics are not inherited in the mitochondrial DNA. So the nuclear DNA will be responsible for how baby look like. But if we know that the oocyte donation will be a treatment option, we of course will share the phenotypical characteristics, but donors are specifically selected. So it will be either very limited option of donors to choose from because these are proven donors who are selected by the medical and the lab team for this technique, but not how they look like. Or also we did this before that we used MRT donor for the purposes of the mitochondrial replacement treatment, and separately we fertilized donor oocytes from the specific donor with the specific phenotypical characteristics, which was chosen by the patient. This is also possible.
Okay, thank you again for that. Let me just go through the next question. As I mentioned, we will be slowly finishing, but the question is, am I correct that you said MRT does not increase euploidy rate, but if you happen to get a euploid, it increases the chance of successful implantation and birth?
It is correct. Neither of the technique will not increase the euploidity rate because it works totally opposite. It creates the environment to increase euploidy. And if you're getting euploid embryo, this euploid embryo will have the highest chances possible for the implantation. For example, we do not have so many surrogacy cases, but it's also possible to proceed with the surrogacy treatment following the MRT techniques. Because embryo is genetically related to the parents, even if this is a specific requirement which is needed, and sometimes we just do not have proper condition for the implantation for patients in their advanced maternal age. So implantation potential will really depend on the environment of the uterus and the quality of the embryo. So from 6 transfers which we transfer to the surrogates, 5 are clinically pregnant and they are carrying the babies. 2, they were already babies were born. So it's very high implantation potential, and we are talking about the most appropriate condition as choosing the surrogate mom. It's never 100%. This is what I said, like we wish it to be and all 6 to be successful, because this is very important embryos for us. As I said, we are taking care about each and every pregnancy because we take care about the technology itself, but we also take care about our patients. So even if you will be our patient without MRT, we will do the best care, of course, for the transfer and the egg retrieval and so on. But we precisely are talking about this technique today.
Of course, definitely. Thank you so much indeed. As I mentioned, we will be finishing for today, but of course, I want to make sure that I will put the email address which you can use, and if you have more, you would like to get more details, and I'm sure there are plenty of your questions that are very personalized as well, of course. So I think it's always best to simply directly contact Dr. Uliana, her team at IFG, because that way they will be able to properly answer as well. So thank you so much. I do see one more question. I still think we can do it. This is a question that okay, I've seen it. Sorry. How do you know for sure that putting my old spindle in a young donor egg with young energy, that it's not helping increase the chance of euploid rate? If you can have a look at this question.
So transferring the spindle from environment of the advanced maternal oocyte, if we reconstruct this oocyte and we put the spindle into the young and healthy environment, taking into account the statistics which we are having, we increase euploidity rate. And we are talking about, in average, euploidity rate of 64% currently. We will be able to distribute this in between the age groups. I know this is a very common question, and you are asking, and what about specifically 47? What about 45 plus? What about 50 plus? What if this is 42 to 43? Because of the numbers, like we do not have a huge numbers. Like if we would divide this into such a small pieces, the variation will be very, very high. So this will be a bias of the small numbers. That's why we are having these details all together. But while we will be increasing the number of patients, those statistics that will be more relevant. So I am happy to prepare these statistics and to share with you, of course. But in general, this is very, so what I can say right now that we do not see the decline on the euploidity rate from 39 to 52. So it doesn't work like this, that in 39 we have the highest euploidity rate, and in 50s we have the lowest euploidity rate. It always goes like this. So one patient would have a higher euploidity rate than another patient, and even if that will be different in the different cycles. We also had patients who completed several mitochondrial replacement therapy rounds, and they had different euploidity rate, but still it was much, much higher, times higher compared to the regular IVF treatments.
Okay. Again, thank you so much. As I mentioned, we will be finishing for today, but thanks everyone for your questions, for your attendance. Lots of you came today for this very interesting topic. Dr. Uliana, thank you so much. It was lovely to have you. And I know we could definitely discuss it more and longer for sure. But I just want to let you know that we will be back with another topic on mitochondrial donation, and Dr. Uliana will also be back. So stay tuned. So you can go to myivfenses.com. All upcoming events are right there. But I can assure you more events on this particular topic, and also with Dr. Uliana will be there. So just please give us some time to prepare everything, and I'm sure it's gonna be another interesting session. Dr. Uliana, any final words before we finish?
Thank you so much, Carolina. And again, thank you for this education and awareness. This is costs a lot, but also for our patients. I know that sometimes to make a decision, it's not easy. And time is an enemy. Because we are talking mostly, I can tell you for sure that 1/10 of our patients are those with the mitochondrial disease disorders. And 9/10, these are the patients who are looking for the solution of inherited their own DNA into their own babies. And I'm happy to support this. I truly, as I said, I truly believe in technology. I see wonderful results and the huge difference among the techniques. And I trust our team. That's why I trust my stimulation, my oocytes to our team, and also as a part of the scientific experiment, which we will share this a little bit later. So I'm happy to be in that category of advanced maternal age because not everyone is going to do it voluntary. So yeah, that's a development, and this is the achievement we are having. So if you are thinking about this, just start acting, and we are here to help you.
Brilliant. Thank you so much. Sounds good, of course. Thank you so much, everyone. Again, it's been a great session. Lovely to have you. Dr. Uliana Dorofeyeva was our speaker today. What an amazing session. So thanks a lot for that. We will be back, so stay tuned. And I wish you all have a great evening. And of course, speak to you soon, Dr. Uliana, as well, right?
Thank you. Bye-bye.
Thank you. Take care. Bye-bye.